Hello, this project was created by Baurmeister and Dedhia. The purpose of our project is to model the synthesis of proteins from amino acids, specifically the first step of protein folding, which is related to the hydrophobicity and polarity of each individual amino acid. We were inspired by our one homework that was about translating bases to their amino acids. We are also inspired by this toolbox, that bioinformatics toolbox that is available through MATLAB, and we wanted to challenge ourselves to make our own. We were also inspired by this specific educational tool, and we wanted to determine the difference of when it's mostly hydrophobic, mostly hydrophilic, as well as the difference that a solvent makes. So the way our code works, you start in the start script, which guides you to run the protein synthesis script, which is here. This script is mostly about figuring out the graphics, what it's going to look like. So the key components really start with these functions. This function ties each amino acid to its specific hydrophobicity, polarity, charge, molecular weight, and pKa. We found all of this information through research, and we use it in our functions that are going to be presented later. This function is very reminiscent of our past homework, which basically takes the 3-digit codon and turns it into what the amino acid is, and creates the amino sequence output, which is used in the following ones. The initial position function is meant to create the string of amino acids and plot them. And then the move acids function takes them from their initial position, takes into account whether it's water or oil for the solvent, and moves them by their hydrophobicity. The more hydrophobic will move faster to get away from the water, or will move faster to spread out in the case of oil. And it is also plotted using the same characteristics as in the initial position. So the protein synthesis script just puts it all together. So, for example, you are also able to change a solvent and RNA sample within protein synthesis. This takes the first RNA sequence that we use as a tester, and the solvent in this particular one is water, and this is the output. It includes the initial, what it would look like, as well as the mass of the protein, the polarity overall, and the pH overall. And you can see we can go in and change to the second RNA sequence and run it again, which gives you a different one, as well as the polarity is shown. And we can also change what the solvent is. We change this to oil here. You can see that it takes a different configuration because what once wanted to be in the center now want to be on the outside. Here's an example of the different way that the protein aligns itself depending on what the solvent is. Thanks for watching. I hope you enjoyed.